This is Dr. Mitch Robinson, and this video is on the Pentose phosphate pathway. It accompanies handout number 9 for cellular and molecular medicine. In this video, we'll examine the cellular metabolism and production of reduced NADPH and pentoses through the pentose phosphate pathway. We'll also examine the role of glutathione in the body and the metabolic disease glucose 6-phosphate dehydrogenase deficiency. On page 2 of your handout, we can see how the pentose phosphate pathway fits into the big picture of intermediary metabolism. The pathway begins at the branch point in glycolysis, glucose 6-phosphate, and the products of the pathway are intermediates in glycolysis. The pathway is also known as the phosphogluconate pathway and the hexose monophosphate shunt. The enzymes of the pentose phosphate pathway are located in the cytoplasm of the cell. The function is primarily anabolic, and it's most active in tissues where there's a large amount of synthesis. This includes liver, adipose tissue, adrenal cortex, testis, and lactating mammary glands. In the pathway, 6 carbons of glucose 6-phosphate are converted to a 5-carbon sugar phosphate, and in the process, reducing equivalents are formed in the form of reduced NADPH. Now, the pentose phosphate pathway has both an oxidative and a non-oxidative branch. Let's look first at the oxidative pathway. In the first reaction, glucose 6-phosphate is oxidized at carbon 1 to a lactone, and reduced NADPH is formed. This reaction is catalyzed by glucose 6-phosphate dehydrogenase. Lactonase then adds water to break the lactone ring and forms 6-phosphogluconate. There's now a carboxyl group at carbon 1, and this can be decarboxylated in the next step. The enzyme is 6-phosphogluconate dehydrogenase. This oxidative decarboxylation reaction yields another molecule of reduced NADPH and a 5-carbon ribulose 5-phosphate. The non-oxidative pathway allows the pentoses that are generated in the oxidative branch to be converted into glycolysis intermediates. All the reactions in this part of the pathway are reversible. There are a series of reactions in which carbon groups are transferred between molecules. You don't need to be concerned with the reactions in this pathway. However, you should recognize that the pathway can take a net 3 molecules of ribose 5-phosphate and produce 2 molecules of fructose 6-phosphate and a molecule of glyceraldehyde phosphate. The key enzymes in the pathway are transketolase and transaldolase. Transketolase transfers 2 carbon groups, while transaldolase transfers 3 carbon groups. The oxidative and non-oxidative branches work together to make the pentose phosphate pathway a multifunctional pathway. For example, the oxidative branch alone can yield both pentoses and reduced NADPH when both are needed in the cell. If only NADPH is needed, then the pentoses can be returned to glycolysis by the non-oxidative branch. The non-oxidative branch can act in reverse to generate pentoses in the absence of NADPH production. The pathway is regulated by the levels of reduced NADPH and pentose phosphates. A high ratio of oxidized to reduced NADPH increases the activity of the dehydrogenase enzymes in the oxidative branch. In the non-oxidative branch, all these reactions are near equilibrium. They're reversible, and so they can simply respond to the level of ribose phosphates in the cell. It should be noted that there are alternative sources of NADPH in cells. For example, isocitrate dehydrogenase is not only a mitochondrial enzyme, but it also exists in a different form in the cytosol, one that uses NADP as a cofactor. So here is an example of an enzyme that can generate NADPH in the cytoplasm. Glutathione is a tripeptide composed of glycine, glutamate, and cysteine. Notice that the glutamate is attached through its gamma carboxyl group, not through the usual peptide carboxyl. The cysteine thiol group of glutathione acts as a reducing agent in the cell, in a reaction where two molecules donate electrons or hydrogens and become oxidized. This cross-linked form of glutathione is the oxidized form. It's abbreviated like this, GSSG. Glutathione exists in relatively high concentrations in healthy cells, and most of it is in the reduced form. This reducing potential of glutathione is used in many reactions in the cell. The large quantity of glutathione in cells is maintained by reducing power generating by the pentose phosphate pathway. The reduced NADPH of the pathway is used to generate reduced glutathione, as shown in the figure on the top of page 6. The key enzyme is glutathione reductase. So this enzyme transfers hydrogens from NADPH to oxidized glutathione. As I said, the reducing potential of glutathione is used in many reactions. For example, it helps maintain the sulfhydryl group of proteins that are in the reduced state. One important site for this is in the erythrocyte, where glutathione maintains the free sulfhydryls so that hemoglobin molecules don't cross-link. It also helps maintain the heme group of iron and hemoglobin in the ferrous state. Glutathione is used in the cell as an antioxidant. It plays an important role in eliminating reactive oxygen species that are formed in the course of aerobic metabolism. This includes hydrogen peroxide and other oxygen species. Glutathione peroxidase uses glutathione to eliminate these damaging peroxides. This enzyme requires selenium for activity. So we need selenium in the diet to carry out this important antioxidant process. Glucose 6-phosphate dehydrogenase deficiency is a genetic disorder in which the first enzyme of the pentose phosphate pathway is affected. People with this disease have a reduced ability to generate reduced NADPH. This is an X-linked recessive disease, and so symptomatic patients are almost always males. Females are almost always asymptomatic carriers. The disease usually remains asymptomatic even in males until there is a situation that causes stress on the antioxidant system. This could be due to things such as infection or the response to several drugs. There's some foods that can also cause an antioxidant challenge. Fava beans, for example, cause a reaction in many people with this disease. The disease has even come to be known as favism in many parts of the world. The main symptom is an elevated rate of erythrocyte hemolysis. And this is usually a hemolytic episode that can lead to anemia, dark urine, and an enlarged spleen. Jaundice, fatigue, shortness of breath can also occur. This hemolytic crisis is usually self-limiting. Now, this figure shows how the defect leads to reduced glutathione and antioxidant capacity in erythrocytes. It's important to point out that erythrocytes do not have an alternative to the pentose phosphate pathway to produce NADPH. There are two enzymes in the pathway that produce NADPH, but since the first enzyme is blocked, you don't have the substrate for the second reaction. Reduced NADPH is needed to maintain reduced glutathione, and when there's an oxidant stress, the low activity of the pentose phosphate pathway can't keep up with this demand. If the amount of reduced glutathione in the cell gets too low, hemoglobin will cross-link and precipitate. This can be seen as dark inclusions in stained cells. These are called Heinz bodies, shown here. This slide shows another characteristic cell from G6PD patients. This is a bite cell. It results when areas of damaged membrane have been removed. G6PD deficiency is thought to be the most common inherited disease in the human population. There are over 400 million people affected. This figure shows the distribution of the disease worldwide, and you can see that it's prevalent in Africa and Middle Eastern European countries. The reason for this distribution and for the widespread occurrence of the disease is that affected individuals have resistance to the malaria parasite, which invades and reproduces in host erythrocytes. And this completes our video on the pentose phosphate pathway.